Inside nearly every complex cell on Earth, something ancient is still at work. Tiny structures moving through the cell's interior, constantly producing energy. These are called mitochondrion. They convert oxygen and nutrients into a molecule called ATP. ATP powers nearly every biological process in complex life. Every animal, every plant, every fungus, all rely on mitochondria to survive. But mitochondria have a strange feature. They contain their own DNA that is separate from the genetic material stored in the nucleus of the cell. Even more surprising, this DNA closely resembles the DNA of certain bacteria in both structure and sequence. This similarity led scientists to a remarkable conclusion. Mitochondria were not always part of complex cells. They began their existence as independent organisms. More than 1 billion years ago, the ancestors of mitochondria were free-living bacteria drifting through Earth's oceans, small, efficient, and unusually good at using oxygen. By this time, oxygen had begun accumulating in the atmosphere after the Great Oxygenation Event. For many organisms, oxygen was dangerous, but for some bacteria, it was an opportunity. These microbes could use oxygen to extract far more energy from nutrients than most cells on Earth. They were, in a sense, biological power plants. At the same time, another type of cell had recently evolved, larger cells with nuclei and complex internal structures. The first eukaryotes. But these new cells faced a problem. Their complexity required far more energy than simple cells could easily produce. Then, sometime in the distant past, these two very different organisms encountered each other, one small and efficient, the other larger but energy-hungry. What happened next would become one of the most important biological events in the history of life, a partnership that changed evolution forever. For most of Earth's early history, oxygen was rare. Life evolved without it, but slowly, the chemistry of the planet began to change. Photosynthetic microbes were releasing oxygen into the oceans. At first, the gas reacted with iron and other elements in seawater. But over time, oxygen began accumulating in the environment. For many organisms, this new gas was dangerous. Oxygen easily reacts with biological molecules, damaging proteins and DNA. To early life, it was often toxic. Many ancient microbes had evolved in a world without oxygen. They had no defenses against its chemical effects. As oxygen spread, entire communities of organisms were forced into shrinking oxygen-free environments. But a few microbes were beginning to do something remarkable. Instead of avoiding oxygen, they were learning to use it. Oxygen makes an extremely powerful partner in metabolism. It allows cells to extract far more energy from nutrients than older chemical pathways. Some anaerobic processes release only small amounts of energy, but oxygen-based metabolism can produce more than 10 times as much. For the microbes capable of using it, this was a massive advantage. Among these organisms were bacteria closely related to modern Alphaproteobacteria. These microbes specialized in oxygen-based respiration. They were small, efficient, and extremely productive energy generators. Inside their cells, oxygen helped drive complex chemical reactions that produced large amounts of ATP, energy that could power movement, growth, and reproduction. As oxygen levels continued rising, these bacteria spread through marine environments. They were well suited to a planet that was slowly becoming more oxygenated. At the same time, early eukaryote cells were also spreading through the oceans. These larger cells had complex internal structures and growing genetic libraries. But they faced a fundamental limitation. Producing enough energy to support that complexity was difficult. Their internal chemistry required far more power than simple cells typically produced. And somewhere in the vast oceans of early Earth, these two very different organisms would eventually meet, one with abundant energy, the other with the potential for extraordinary complexity. Their encounter would reshape the future of life. Early oceans were crowded with microscopic life. Cells constantly collided, competed, and sometimes consumed one another. Unlike bacteria, many early eukaryotic cells could surround and engulf other cells. This process is known as phagocytosis. Normally, the engulfed organism would be trapped inside a membrane bubble, then broken down and digested. But in one rare encounter, something different may have happened. A small oxygen-using bacterium was swallowed by a larger host cell. Instead of being destroyed, the bacterium survived, protected inside the host's internal environment. Inside the host cell, the bacterium continued producing energy through oxygen respiration. Large amounts of ATP, far more than the host cell could normally generate. Some of this energy began benefiting the host. The larger cell gained access to a powerful internal energy source. In return, the host cell offered something valuable to the bacterium: protection and a steady supply of nutrients. This type of biological partnership is called endosymbiosis, one organism living inside another in a mutually beneficial relationship. As the host cell reproduced, the bacterium divided as well, passing from one generation of cells to the next. The partnership continued. Over many millions of years, the relationship grew deeper. The bacterium began losing genes that were no longer necessary for independent life. Some of its genes even moved into the host cell's nucleus. Gradually, the once independent bacterium became more and more integrated with its host. What had once been a free-living organism was becoming something new, a permanent structure inside the cell, the first mitochondrion. Once the partnership stabilized, something extraordinary happened. Cells suddenly had access to enormous amounts of energy. Inside the new mitochondrion, oxygen-driven metabolism produced ATP far more efficiently than earlier chemical pathways. ATP is the molecular fuel that powers living cells. Cells without mitochondria must produce energy across their outer membrane. But mitochondria brought thousands of internal energy-producing membranes into the cell. The inner membrane of mitochondria forms intricate folds called cristae. These folds dramatically increase the surface area available for energy-producing reactions. Within these membranes, oxygen helps drive a process known as cellular respiration. This system extracts large amounts of energy from nutrients like glucose. With this new energy supply, cells could maintain larger genomes, build complex internal structures, and control increasingly sophisticated chemistry. Over time, eukaryotic cells became larger and more capable. They could move, capture food, and interact with their environment in new ways. These cells began evolving into many different forms. Some specialized in hunting other microbes. Others absorbed nutrients directly from the water. Eventually, some lineages began experimenting with something even more unusual: cells remaining attached after division. Small communities of cells working together. This was the earliest step toward multicellular life, a development that would eventually produce organisms large enough to be seen with the naked eye. All of it powered by the energy made possible through the mitochondrial partnership. Every complex organism alive today still depends on this ancient alliance. But the story of mitochondria did not end with their origin. More than 1 billion years after the first partnership formed, mitochondria are still inside our cells, quietly doing the same job they began long ago. Unlike most structures in a cell, mitochondria can reproduce on their own. They divide in a way that strongly resembles bacterial cell division. They also carry their own genetic material, a small circular genome that remains separate from the DNA stored in the nucleus. This genetic evidence helped confirm what scientists had suspected. Mitochondria were once independent bacteria, relatives of modern Alphaproteobacteria. Every animal cell contains mitochondria. So do plant cells and fungal cells. Across nearly all complex life, the same ancient system continues to operate. Without the energy produced by mitochondria, complex cells could not function. Their internal chemistry would simply require more power than the cell could generate. This single evolutionary partnership made it possible for life to grow larger, more complex, and more diverse. It helped open the door to multicellular organisms. Over hundreds of millions of years, that complexity would eventually produce forests, coral reefs, and entire ecosystems of visible life, all powered by descendants of a bacterium that once lived freely in ancient oceans. Inside every cell of your body, that ancient partnership continues. A reminder that some of the most important events in the history of life began with something almost invisible, one cell learning to live inside another. For more on early life on Earth, please like, subscribe, and ring the bell. This has been For What It's Worth.